Hi everyone, we're on our last video for activity 2, and I think that this is probably the most important video for your report sheet of any of the videos of this whole series, because this will really help you to answer your questions in the report sheet. So we're talking about the tonicity differences between plant and animal cells. And this goes back to the vocabulary that we were talking about. Tonicity is hypertonic, hypotonic, isotonic. So let's talk about animal cells up top and plant cells on the bottom, and the differences in how tone impacts these two cells or types of cells. So in an animal cell up top, that could be a red blood cell. And if it is in an isotonic environment where the concentration of solutes inside the red blood cell and the concentration of solutes in the extracellular solution is equal and has equal concentration, then you're going to have a homeostatic environment where water can come into the red blood cell and water can go out of the red blood cell and come in and go out and come in and go out. There's not much of a gradient there, and so it's going to be a very slow movement in and out, but these are isotonic solutions that are in equilibrium in an animal cell. And this is normal for an animal cell. Not the case with plant cells. Isotonic solutions for plant cells don't go over well. Here's what's going on. When you have that slow movement of an isotonic environment, the plant is not happy because the plant wall and the plant membrane don't have pressure behind them to fill out the cell. As a result, the cell leaf will wilt, actually. So let's go into that a little bit more. An isotonic environment for a plant cell means that there's little tendency for water to come into the cell. That cell wall is rigid and has now a plasma membrane nearby that isn't pushing on it and adding extra rigidity to the cell wall. Without that extra rigidity and that extra support from the plasma membrane, it can result in flaccid cells and wilted leaves. So you get the idea that plants are very susceptible to dehydration. Let's talk about another situation. Let's talk about a hypotonic environment for a red blood cell. So that means that there's hypotonic extracellular solution. Maybe you drank an entire glass of pure water and now your stomach cells are dealing with the influx of this pure water that has no solute in it. So you have a hypotonic extracellular solution coming into your body without any solute in it. It's hypotonic, means that the cell is relatively inside hypertonic. It has solute inside its cell. It is hypertonic relative to the extracellular solution, which is flush with water, and hypotonic. In that case, you know that the hypertonic environment will take in water along its semi-permeable membrane, especially when the solutes can't move well, and a hypertonic solution will take in water into the cell and swell, and as it swells, that plasma membrane may burst and lyse the cell, burst the red blood cell. That's what will happen in a hypotonic extracellular environment for animal cells. Let's see what happens in plant cells when they have a hypotonic extracellular solution. Well, what would that be? That would be very clean rainwater in this case. So sometimes rainwater comes in and it has dust with it, and sometimes rainwater comes in and it has salt spray from the ocean with it, and sometimes rainwater comes in and it has acid in it for acid rain. But if we have really clear rainwater that has no solute in it at all, it is going to be a hypotonic solution for that plant cell. that means that the plant cell inside of its cell is hypertonic. And that hypertonic intercellular environment is going to take in water. That plant cell is going to take in that pure rainwater. And when it takes in that pure rainwater, the plant cell will swell, but it's got a cell wall with some real structure to it. So when that plant cell swells, it swells and the plasma membrane pushes on the cell wall to give it some support. That is a situation that the plant wants to be in. We call that turgor pressure. And it is the normal situation for a healthy plant in that the osmotic pressure is high enough inside the hypertonic cellular environment so that the cell wall accommodates the swelling, but then starts to oppose any more water coming in because it's got enough. And that is what we call turgid pressure for a healthy cell plant. That's what we want to see. We want to see turgor pressure within the plant cell. It means that the plant cell wall has actually got so much that it's just pushing back. No more, please. No more. We're full. Another situation which you commonly find is when you have a dehydrated body or a dehydrated plant, really severely dehydrated. And I deal with this with EMS all the time. Dehydration is one of the major reasons for 911 calls. So in that case, that means that the extracellular solution around the red blood cell is hypertonic. It has a lot of concentrated solute in it because the person is dehydrated and just doesn't have enough water in their blood. In that case, the blood wants to take water in because it is a hypertonic solution. It's taking water in from the cellular environment. Removing water from the cellular environment shrivels the cell. It can shrivel the cell up to the point where the cell is very ineffective. And that is a major reason for 911 calls for me. Obviously, it's a detrimental environment when you have a hypertonic solution around a red blood cell. On the other hand, with plants, let's talk about what happens there when they have a hypertonic solution around them. Maybe they have salt spray in from the ocean with their rainwater. Maybe they have a lot of dust in their rainwater. Maybe they have acid rain and acid solutes in their rainwater. That makes the rainwater that this plant cell takes in into their roots, that makes that a hypertonic solution. Well, in that case, the osmotic pressure is on that hypertonic solution is to take water in from plant cells in order to try to equilibrate that hypertonic condition. In that case, that water taken in from the plant cells really shrivels the plant cell to the point where the plasma membrane can pull away into the interior of the cell, away from the cell wall. That is called plasmolysis. In other words, lysing or splitting the plasma membrane, leading to cell death. So there are conditions that are involved in animal cells and plant cells. The conditions are the same, isotonic, hypertonic, hypotonic, but the clinical outcomes are different, and you'll need to do that and examine that on your report sheet. So I hope that this will help you, and you can refer back to these slides. See ya.